Good evening to one all present here. Thank you so much for making it on time. Welcome to Lifeline Connect by Deborah Rice Movement, or what people popularly call it as DRM. Our motto at DRM is to give voice to the voiceless, to advocate for the broken, and rescue the abused. You can find out more about us on www.deborahrice.org. We have free counseling sessions, and if you can reach us through our website or through the number that is 8801-858585. So I'll be putting that number on the chat in a while, so you can contact us via that number. So today we are touching on a very important topic, that is scientific challenges to neo-Darwinism. So the first episode is already over, and today we have the second episode on Cambrian Explosion. So with us today, we have three speakers. Dr. Lata Christie, a senior scientist from Bangalore, India, with more than 33 years of experience. After her BE with honors, she completed her master's and PhD in aerospace engineering from the Indian Institute of Science, Bangalore. She is the author of 4 books, and she has around 70 publications in peer-reviewed journals and conferences. She has received many awards and has completed her master's in divinity from SABC, Bangalore. She is also trained in apologetics and counseling. Dr. Jonathan Mcclatchy is a Christian writer, international speaker, debater, assistant professor at Sattler College, Boston, and a fellow of the Discovery Institute. He's earned a PhD in evolutionary biology from the Biosciences Institute at Newcastle University in the United Kingdom. Prem Isaac worked in software development and data science for 29 years with undergraduate degrees in physics and computer sciences. He has an MA in philosophy as well. He has also served as the international director for Ratio Christie. Prem has taught New Testament and apologetics at churches and colleges. I thank each one of you who are with us today, and God bless you all. I'd request Dr. Lata Christie to take over.
Thank you, Disha, for that introduction. Today, we are going to deal with a very important question, the question of Cambrian explosion. A mere 542 million years ago, the Earth's landscape was a wilderness. Many million years after our planet Earth had come into existence, when there were no animals on the land, suddenly something extraordinary happened in the ocean bed. Over a period of 10 to 20 million years, or the beginning of a period called the Cambrian, explosion of new animals developed below the water surface. These gave rise to most of the diversity of animal life that now exists on the Earth. Cambrian explosion is a term that refers to the geologically sudden appearance of most of the major group of animals. Many species in several domains of the animal kingdoms appeared at about the same time geologically. The fossil record shows the appearance of many living phyla for the first time, and this gives evidence for the sudden emergence of animals and the variable explosion of life. So most geologists date this Cambrian explosion to around 530 to 520 million years back. The term explosion is given because it's not an explosion in terms of animals and birds flying all over the place. Actually, when we talk about an explosion, what the geologists mean is effectively an enormous diversification, the abrupt appearance of animals and a lot of different types of organisms. But the cause of the sudden burst of life, which geologists call the Cambrian explosion, is still now remaining a great mystery. The explosion was really life's Big Bang. Things went from moving very slowly along the seabed without any predation to suddenly all the different types of ecologies that we find today appearing. Most of the major forms of animals appeared suddenly in a short span of time. From nothing, we have almost everything. Your life literally exploded. That's a great mystery, and no one can understand how this happened. But how did this all start? How people found out about this Cambrian strata? In 1831, Charles Darwin joined a life-changing expedition. But before going on this voyage on board the HMS Beagle, the 22-year-old Darwin was introduced to 46-year-old Adam Sedgwick, a professor of geology and botany at Cambridge University. Since Sedgwick was studying the geology of whales, he invited Darwin to join him at a field trip from Shrewsbury, Darwin's hometown, to the island of Anglesey. Sedgwick would later use all his collected observations to introduce the geologic epoch called Cambrian. This epoch was named after the ancient Roman name for whales. Since Darwin was interested to acquire the basic skills of geological field research, he assisted the renowned geologist Adam Sedgwick. For Darwin, the fossils embedded in the Cambrian shale was a curiosity. That incident intrigued Darwin, and down the line, he imagined that natural selection and the whole theory of evolution, and wrote the book, The Origin of Species. However, after so many years later, he could never resolve the mystery he was trying to resolve. That's the Cambrian explosion. And then later he passed on the mystery to future generations. Today, most paleontologists say that complex animals first appeared around 530 million years ago during a short span of time. Let me share my screen and present. Initially, the land was totally barren, no trees, no plants, no evidence of bacterial life also. But on the other hand, the sea was teeming with life. Around 530 to 520 million years back, plant and animal species suddenly appeared from nowhere with great diversity. That's what Cambrian explosion is all about. You could see so many species here. All of them came at one time. You can see in the same strata in the fossil record. So this is a real mystery. Why it's a real mystery? Because it's not only a mystery, it's a challenge to Darwin's theory of evolution. Because all these things happened in a short period of evolutionary time scale. And the development of complex animal forms came up in a burst. Because as per Darwin, there should be a gradual evolutionary process. So we should find a lot of predation to this animal forms, but nothing were there earlier. Suddenly everything came into existence. And as per Darwin, there should be a lot of transitional forms, but nothing is there. And that is another mystery of missing transitional forms. And Darwin said that the base of the tree of life should be a simple organism, and then it branching into a great diversified form of life with human life appearing on the top. But now it looks like upside down, the bottom of it looks. So these are all some of the challenges to Darwin's theory of evolution. You can see here, this is a trilobite. These are some of the things that appeared in the Cambrian strata. So you can see this trilobite. It looks a lot like a crab, present crab, shell with legs, with mouth, with compound eyes, and many things else. So how this came into existence? This is a shale that they discovered, Burgess Shale. And in this one, they found a lot of Cambrian deposits. And this brought in a lot of issues to Darwin theory of evolution, because you can see suddenly shells forming, you can see compound eyes, you can see many forms of great higher animal life existing at the same time along with simple animal forms. So everything existed together. So this is a great mystery. So in this fossil deposit was found in Western Canada. It provided the first example, but now from China and many other provinces, we can find the Cambrian strata with a lot of different type of animal forms, with just greatly developed animal forms. So it's called as an explosion because it happened within a few million years. And this is truly an explosive event in the scientific terms. What we are seeing is a quantum jump. And this quantum jump has no explanation at all. And this, because of this quantum jump, the gradual theory of evolution proposed by Darwin has become a great question mark, because it's no more a tree of life like this. It's totally different. It has become a bush. So now what are the other challenges? The shorter lifetime. It takes a small percentage of time in the geological history to develop so many bigger animal forms. There is so much of innovation in such a small fraction of time that no one expected, because on the Darwinian basis, you can see mechanism of natural selection acting on random variation. You will have small incremental changes, and to develop a complexity, it takes a long time. So in the Darwinian scale, 10 to 20 million years is just a blink of the eye. So it's just a blink of the eye, everything happened. Why it's a blink of the eye? There is a branch of evolutionary theory known as population genetics. It's a mathematical branch of biology in which biologists are able to calculate what they call as waiting times. We discussed about this in the first episode. This is the amount of time that you would expect for a given evolutionary change to take place. And biologists can make this calculation if they know something about the mutation rate, the generation time, the size of the population of different organisms that are involved in that. But in recent times, with this population genetics, to get a few coordinated mutations, the waiting times blow up beyond hundreds of millions of years. And so to get a small good mutation, you need a lot of time. So trilobites with this compound eyes that can actually be seen in the fossils would have taken a lot of time. But something amazing, that time was not involved. Everything came in a burst. So that is a great mystery. So this mystery no one could solve. And again, if you see, when Darwin looked at Galapagos Islands and the finches and their beaks, it was just a gradual process. Even if you see the breeding and generation of breeding experience with breeders, you can see the pigeon breeders are performing in their pigeon coves and dog kennels, and even the sheeps, to make it more woollier sheeps, you try to breed with a more woollier sheep so that the next generation will become more woollier. So that is adaptation. That is not an evolution. But to make a great change from one thing to another, you need a lot of coordinated mutation. And for that, 10 to 20 million years is just a short span of time. And even in that fossil record shows very sudden appearance of all the animals. There is no transitional forms involved. We would be speaking about the fossils, the transitional forms, and the dating approach in our next episode. So this time, I would like to tell you, try to search for the transitional forms from the simple organism to the complex organism. You will not be able to find in the Cambrian strata. So that's why evolutionary trees no more a tree of life with the root and then going up into branches. It's just a bush. So now with all these things, let me ask a question to our guest, Jonathan. Jonathan is here with us. And it's a great privilege for us to have Jonathan here. He's an evolutionary biologist. And so let me ask my question to him. So some of the defenders of Darwin's theory argue that random mutations in a special set of genes called the Hox genes are responsible for dramatically speeding up the evolutionary process through the Cambrian. But what's interesting to me is these genes are turned on only long after the body plans were established. So Jonathan, would you throw some light on this, and could you explain what is this Hox genes and all these about, and how evolutionists are trying to explain this? Over to you.
Okay, well, thank you so much for that great introduction. My PowerPoint on the origins of animal body plans. So thanks to Dr. Christie for giving us an excellent introduction overview to the challenge of the Cambrian explosion, to the evolutionary narrative in terms of the origins of the animal phyla, which are the body plans of organism, which, as Dr. Christie said, emerge within a very abrupt window of geological time, about 10, 20 million years. So what I want to do today then is to analyze or to give a basic overview, introduction to what it takes in terms of at the molecular level to produce a body plan, and how development works. I'll give a short overview of developmental biology and some of the challenges that that pertain to the Cambrian explosion in terms of the origins of novel body plans. So I want to begin then with discussing the field of evolutionary developmental biology, which is often known as EVO-DEVO for short, and how this relates to the case for intelligent design. So, multicellular development in plants and animals follows a body plan or pattern. Body pattern refers to the spatial arrangement of different body regions. So at the cellular level, the body pattern is due to the arrangement of cells and their specialization. The progressive growth of a fertilized egg into an adult organism involves 4 types of cellular events. We have cell division, cell migration, cell differentiation, and cell death. So here is a figure which shows the different types of cellular events that occur during the course of development. So we have cell division, we have cell migration, we have cell differentiation, where you go from an undifferentiated cell to a differentiated cell, which is committed to performing a certain role within the organism's body, such as a nerve cell in this case. And then we also, of course, have cell death or apoptosis. And apoptosis is a programmed or organized form of cell suicide. And actually, apoptosis is very important in the development, especially of our digits. So if you look at your fingers and toes, these were actually carved out by programmed cell death or apoptosis. And this actually by itself is a significant challenge to the evolutionary narrative, because it's very difficult to envision by an evolutionary mechanisms how you would find those how you would evolve such a coordinated program of cell death in order to carve out digits in a trial and error type of way. It's very surprising that evolution would have selected programmed cell death without foresight as to where that was leading. So each cell of the body has to receive signals or positional information that affects its course of development. A class of molecules known as morphogens are molecules that convey positional information and promote developmental changes. So a morphogen basically influences the developmental fate of a cell. And a key feature of morphogens is that they act in a concentration-dependent manner. So a particular concentration range of one or more morphogens restrict a cell into a specific developmental pathway. So within an oocyte, which is an egg cell, morphogens may occur along a concentration gradient. And as you can see here, this is a morphogen at the interior end of this particular oocyte. And morphogen gradients are also established by secretion and transport to neighboring cells, as you can see in this diagram here. So the process by which a cell or a group of cells governs the developmental fate of neighboring cells is known in developmental biology as the process of induction. Positional information is also conveyed by cell adhesion. So each cell makes its own collection of cell surface receptors, and these are known as cell adhesion molecules, or CAMs for short. And these cause the cell to adhere to the extracellular matrix and or to other cells. The study of mutations has greatly contributed to our understanding of development. So, for example, a mutation in a complex of genes called bithorax in Drosophila melanogaster, which of course is the famous fruit fly, causes the fly to have 4 wings, have an extra pair of wings, an extra set of wings which replace the balancers, the halteres that stabilize the organism during flight. And of course, unfortunately, the Drosophila with its extra pair of wings is a hopeless cripple. They can't survive outside the laboratory because the extra set of wings are not connected to muscles or the brain, and so they're not actually able to fly, and they hinder the organism rather than help it. But this is an example of the sort of mutation that you can induce. in this particular complex of genes. And genes that specify the final identity of a body region are known as homeotic genes or Hox genes. And they're defined by a region known as a homeobox region. So Hox is short for homeobox. Most animals, including ourselves, are bilaterians with left-right symmetry. So we're essentially symmetrical, left to right. And development in bilaterians generally proceeds in 4 overlapping stages. So you have the formation of the body axis, as you can see here. So you've got the anterior, posterior, and dorsal ventral axes of the organism. And then the segmentation of the body, as you can see here, is divided into segments. And then determination of structures within segments, as you can see here. And then cell differentiation, such that the cells divide and give rise to, so the fertilized egg becomes 2 cells, and then 4, and 8, and 16, and 32, and then 64, and so forth. And at each step, you are changing the regulatory pattern such that different genes are turned on or off, and that determines cell differentiation, what cells are going to be committed to performing certain roles within the organism. And every cell in our body, by the way, has exactly the same genetic code, has exactly the same DNA sequence. It's only a matter of which genes are actually turned on or off in that particular cell type. That's what differentiates, say, a heart cell from a neuron, or from a lung cell, or epithelial cell, or what have you. So the first stage in Drosophila embryonic pattern development is the establishment of the body axis. The oocyte is the cell most critical, as the egg cell, is the cell most critical in determining the pattern of development in the adult organism. So it's an elongated cell with pre-established axes, and these axes are created as certain gene products are deposited asymmetrically within the egg. After fertilization, these gene products establish independent developmental programs. So these govern the formation of the body axis of the embryo. So examples are or genes like bicoid and nanos and torsal and tall. So these gene products act as key morphogens or receptors for morphogens. So then you have the formation of the syncytial blastoderm. So you have a series of nuclear divisions which are not accompanied by a cytoplasmic division. And these nuclei migrate to the periphery of the cytoplasm, as you can see here. So these are the syncytial blastoderm nuclei. And then these cells form, creating a cellular blastoderm, as you can see in this diagram here. Dramatic changes occur to the organism during gastrulation, as you can see here. So this is after about 3.5 hours. So the mesoderm and endoderm are forming from invaginating cells, and that is the ectoderm. Okay, so the embryo divides into units. Initially, shallow grooves partition the embryo into 14 power segments, and these grooves disappear, and new boundaries are formed that divide the embryo into morphologically discrete segments. So this diagram here shows the segmented pattern of the embryo approximately 10 hours after fertilization. So so there are 3 larval stages that are separated by molts, and the larva sheds its cuticle, and then metamorphosis occurs. So a group of cells are known as imaginal discs, which were produced earlier in development, grow and differentiate into adult structures. And then the adult fly emerges from its pupal case, as you can see here, with its anterior-posterior axis, which is left to right, and its dorsal ventral axis, which is top and bottom. Then you've also got the left to right here, and then the proxima distal axis. Okay, as you can see here. Okay, so the maternal effect gene products establish the anterior, posterior, and dorsal ventral axis of the organism. So these are deposited in regions of the oocyte. So the bicoid messenger RNA is distributed in the anterior end of the oocyte, and that promotes the formation of the interior structures, right? So there's a concentration gradient. The nanos mRNA is localized to the posterior end and promotes the formation of the posterior structures. And then you've got the torso receptor, which is shown here, which is found in the plasma membrane and is activated by ligand binding at either end of the oocyte, and that causes the formation of structures that are found only at the ends of the organism. And then you've got the toll receptor protein is activated by the ligand binding at the ventral side of the embryo, and that establishes the dorsal-ventral axis of the organism. So the Bicoid gene gets its name because a larva whose mother is defective in this gene develops with two posterior ends, as you can see here. So this is how it should be with the working Bicoid gene. And you've got the spiracle at this end, whereas if this Bicoid gene is non-functional, then you get two posterior ends of the organism with a spiracle that you can see at both ends. So that's not the way it should be. So a female with one or two copies of the functional Bicoid gene produces a larva that are morphologically normal, and a female fly that's homozygous for an inactive Bicoid allele produces all abnormal offspring, even when the female is mated to a male that is homozygous for normal Bicoid allele. So in other words, the genotype of the mother determines the phenotype of the offspring, and this occurs because the Bicoid gene product is provided to the oocyte via the nurse cells. So in the ovaries of female flies, the nurse cells are localized asymmetrically towards the interior end of the oocyte. And this side becomes the interior of the embryo. And the Bicoid gene is actively transcribed in the nurse cells. The Bicoid mRNA, the messenger RNA, enters the interior end of the oocyte and gets trapped there. Okay, so the next developmental process after axis formation is the organization of the embryo into segments. So this positional information will be maintained or remembered throughout the rest of development. And early in development, the segmentation genes play a role in the formation of body segments. So in the 1970s, segmentation genes were identified by these two researchers, Christie, Nussle and Volhard, and Eric Weishaus, who undertook a systematic search for mutations affecting embryonic development in Drosophila. And their pioneering effort identified most of the genes required for the embryo to develop a segmented pattern. And they found that there are three classes of segmentation genes. There are the gap genes, parallel genes, and segment polarity genes. So the sequential expression of genes divides the embryo into segments. So first, the maternal effect gene products such as Bicoid messenger RNA are deposited asymmetrically in the oocyte, and these form a gradient that will later influence the formation of axes. And then after fertilization, maternal effect gene products activate zygotic genes. So the first set to be activated is the gap genes. And then the gap genes and maternal effect genes then activate the pair rule genes, and the pair rule genes then regulate the segment polarity genes. And then later in development, the interior end of one power segment and the posterior end of another power segment will develop into a segment. And each segment will have particular morphological characteristics. So that's just a summary of what I just said. So the term cell fate describes the ultimate morphological features of a cell or a group of cells. So in Drosophila, the cells in each body segment have their fate determined very early in embryological development, long before morphological features become apparent. So the term homeotic refers to mutant alleles in which one body part is replaced by another. It's coined by William Bateson. So for instance, you can have the halteres, as I mentioned earlier, the balancers to stabilize the organism during flight, replaced with the wings. That would be an example of that. So the homeotic genes are found in two complexes in the Drosophila fly, known as antennapedia and bithorax. And this is actually color-coded to show the part of the body that these genes correspond to. Similarities between the fly and the mouse also suggest that a common plan of body development is found in all animals with bilateral symmetry. So there's 13 different types of homeotic genes that are found in mouse, although none of the 4 Hox complexes actually contains all 13 of those. This shows this is an illustration of Hox gene expression in the embryo and the corresponding regions of the adult. Okay, so let's turn our attention briefly to Hox genes and whether they can help with explaining origins of new body plans and anatomical features. So Hox genes in vertebrates determine the pattern of anterior-posterior axis. During evolution, increases in the number of Hox genes have an important effect on animal morphology. So as an example, differences in position expression of HOXC6 correlate with the number of neck vertebrates produced. So this is a diagram that shows in different organisms, mouse, chicken, goose, and snake, the Hox genes that are responsible for different anatomical structures, such as the hand and a wing and and so forth, the vertebra of a snake. Okay, so the role of homeotic genes has been revealed by mutations that alter their functions. So, for example, the antennapedia mutation in Drosophila is a gain-of-function mutation, and it causes the gene to be expressed in an additional place in the embryo. In that case, it's also expressed abnormally in the anterior segment that normally gives rise to the antennae. And investigators have also studied many loss-of-function allelic homeotic genes. So in particular, homeotic gene is defective, its function is replaced by the gene that acts in the adjacent anterior region. So this is a picture of what happens when the antennapedia gene gets mutated, where instead of having the antenna grow out of the head, you have legs growing out where the antenna should be. So this mutation implies that has a gain-of-function mutation, in which the antennapedia gene is expressed in the embryonic segment that normally gives rise to the antennae. And that the expression of antennapedia causes that region to have legs rather than the antenna. Okay, so the homeotic genes are regulated in a complex way. So you have the polycomb genes, which repress the expression of homeotic genes in regions of the embryo where they should not act. And you have so this is done by remodeling chromatin into a closed conformation so that they can't be as readily expressed. And then the trithorax genes promote the expression of homeotic genes in regions of the embryo where they should act. And that's done by remodeling chromatin into an open conformation. So overall, the concerted action of many gene products causes the homeotic genes to be expressed in the appropriate regions. As I mentioned, the homeotic genes encode transcription factors, which regulate expression of other genes, and genes contain this 180 base pair consensus sequence known as the homeobox. And the protein domain encoded by the homeobox is known as a homeodomain, as you can see here, it binds to DNA. So in terms of evolutionary biology, so this has caused a lot of excitement in the evolutionary developmental biology field. So Jeffrey Schwartz from the University of Pittsburgh in Pennsylvania, he says, we're still in the dark about the origin of most major groups of organisms. They appear in the fossil record as Athena did from the head of Zeus, full-blown and ready to go, in contradiction to Darwin's depiction of evolution as resulting from the gradual accumulation of countless infinitesimally minute variations. A mutation affecting the activity of a homeobox or Hox gene can have a profound effect, such as turning larval tunicates into the first chordates. Clearly, the potential homeobox genes have for enacting what we call evolutionary change would seem to be almost unfathomable. I'm not, though, convinced that you can that this actually is a viable way to explain the origins of novel anatomical features and new body plans and so forth by mutating these Hox genes. For one thing, precisely because Hox genes coordinate the expression of so many other different genes, experimentally generated mutations in Hox genes are proven harmful. Even so you've seen these gain-of-function mutations with the legs coming out where the antenna should be in the Drosophila, or the extra pair of wings in the four-winged fruit fly. But these, although they're gain-of-function mutations, are actually harmful to the organism. In fact, experimentally generated mutations in Hox genes are uniformly harmful to the organism. So the antennapedia mutant fruit fly with legs growing out of its head where the antenna should be cannot in fact survive in the wild. Hox genes in all animal forms are also expressed well after the body plan is established. So although they are expressed early on in development, it's relatively late in early development. So after the body plan, the anterior, posterior, and dorsal ventral axes have already been established. So in fruit flies, by the time that Hox genes are expressed, roughly 6,000 cells have already formed, and the basic geometry of the fly is already well established. And thirdly, Hox genes only provide information for building proteins that function as switches that turn other genes on and off. They don't contain information for building those structural parts. So mutations in Hox genes do not have all the genetic information necessary to generate new tissues, organs, and body plans. So mutating them can't possibly generate new forms of animal life. Furthermore, there have been various studies that have been done on model organisms, including Drosophila, and zebrafish and so forth, C. elegans roundworm, that shows that early acting developmental mutations are uniformly deleterious, in fact, usually lethal to the organism. So this results in a paradox, namely the proposal that early acting developmental mutations can cause stable hereditable large-scale changes in animal body plans contradicts the result of 100 years of mutagenesis experiments on organisms such as fruit flies, nematodes, zebrafish, and mice. So in order to get new body plans, because the body plan is laid down so early in development, you have to have these early acting developmental mutations, but these are the least likely to be tolerated by evolution. Because early acting body plan mutations, because all subsequent development flows from those early processes, invariably generate embryonic lethals, that is, dead animals incapable of further evolution. And in order to generate a genuinely new body plan, early embryonic changes have to take place. So this results in this dilemma of major but not viable, or viable but not major, that the types of mutations that we need to generate new organisms uniformly result in these embryonic lethals, but the type of mutations that we do get aren't the sort of mutations that we need to generate new body plans. So that is a significant problem for the theory of evolution, I think. So I'll pause there and see if there are any questions on that so far.
Thank you, Jonathan. As you said, that Hox genes cannot explain the sudden origin of Cambrian animals that came out of nowhere. Cambrian explosion is not just a problem of explaining the absence of evidence in the fossil record, but it is also a problem of explaining everything we know about life, right down to the level of molecules and cells, as Jonathan spoke. The biological structure of a Cambrian trilobite, if you can see, it is so complex and sophisticated as a modern crab. So its organ, including the brain, gut, heart, and the compound eyes, each organ had to be constructed from specific types of cells that has to be made from protein molecules. And each protein was assembled from a 4-letter chemical code in a section of DNA called a gene. So now, if you would like to explain all these things through evolutionary process that can transform a simple pre-Cambrian organism into a Cambrian trilobite with at least 10 to 20 times more complex, it's a huge leap. And to make that leap, you need a vast amount of new genetic information. So now the question arises, where does the genetic information come from? That is the central mystery of Cambrian explosion. So now, if you look at it, a remarkable chapter in the history of life still wait resolution. So it certainly has the appearance of an intelligent design behind all these things. That's what I could conclude. So I think to build an animal kind of complexity requires a process that can look into the future, that has a foresight, that can bring everything together to actualize something like a trilobite or a mollusk or something like in what we can see in a Cambrian explosion. So it needs to be a process that has a foresight. It has to be a process that can visualize complexity, that has an intelligence, and it cannot be a natural selection, but design. That is my inference. So let me ask a question to Jonathan. Can a process like a blind selection that lacks foresight produce a cell lineage that can culminate in a viable adult roundworm? Can you explain that?
Excellent question. So this brings me on then to a second major obstacle to explain the origins of new body plans, and this is the roundworm C. elegans, incidentally. Okay, so let me go on. Okay, so the life cycle of the C. elegans is the embryo develops within an eggshell, and it hatches when it reaches 550 cells. After hatching, it passes through 4 successive molts in about 3 days. So it's about 3 days for a fertilized egg to develop into an adult worm. And The males produce sperm, and an adult male is composed of over 1,000 somatic cells. And there's also the hermaphrodites, which produce both sperm and eggs. And the adult hermaphrodite is a little under 1,000 somatic cells. So remarkably, the pattern of cellular development in that organism is invariant from worm to worm. And C. elegans is a great model organism for studying development because it is transparent and has relatively few cells. And so you can actually observe, you can actually follow the cell division step by step. And this can be represented with a cell lineage diagram. So you go with you go from the fertilized egg. The fertilized egg will differentiate and divide into 2 daughter cells. And then those will each divide into 2 daughter cells. So that's 4 in total. And then those will all divide into 2 daughter cells. So now you've got 8, and then 16, and 32, and 64, and so forth. And so you can see this cell lineage, and you are, with each round of cell division, changing the expression patterns such that you are, the different genes are being turned on or off depending on what cell type is going to give rise to in that particular organism. So some of those differentiated cells, for example, will contribute to the nervous system, the skin, the musculature, or the gonad, or the intestine, for example, or the germline. Each of those cells will have exactly the same genetic instructions, but different genes will be turned on or off. Different genes will be expressed depending on which cell type you are. And so in order for that sort of process to happen, one of the most vexing challenges, I think, to the evolutionary paradigm is, how do you produce a cell lineage like this without foresight of where you're going? Because at each round of cell division, you are changing the expression patterns. You're then regulating certain genes, not with respect primarily to what those cells are doing, but rather to what the final cells in the adult organism are going to be doing. So you have an end goal, if you will, that you're trying to reach by changing those expression patterns, and that requires a process with foresight. Obviously, it's no use having an organism that only makes it halfway through development and is non-viable. You have to be able to reach that end target, these different cell types require foresight to visualize as a complex end goal and then change the expression patterns in order to arrive at that end goal. And it's very difficult to imagine any process that doesn't have intelligence or foresight being able to bring about that type of effect. So let's then talk about development at the cellular level. So we have so cell determination is defined as when a cell is destined or determined to become a particular cell type. Cell differentiation is when a cell's morphology and function have changed, usually permanently, into a highly specialized cell type. So at the molecular level, the profound difference between cell types arises from gene regulation. So though different cells contain the same set of genes, they regulate the expression of their genes in different ways. So researchers have identified specific genes that cause cells to differentiate into particular cell types. And these genes trigger undifferentiated cells to differentiate into the proper cell fates. In 1987, Harold Weintraub and his colleagues identified a gene which they called MyoD. This plays a key role in skeletal muscle cell differentiation. So when a cloned copy was introduced into fibroblast cells, they differentiated into skeletal muscle cells, whereas normally in vivo, they never actually follow that pathway. So researchers later found that MyoD belongs to a group of 4 genes that initiate muscle development. The other 3 are myogenin, MY5, and MRF4. And all 4 genes encode transcription factors that contain a basic domain and a loop helix domain. And it binds to DNA and activates skeletal muscle specific genes. And it's necessary for dimer formation between transcription factor proteins. The 4 genes are called myogenic BHLH proteins, and they're found in all vertebrates and even some invertebrates such as Drosophila and C. elegans. and they are activated during skeletal muscle cell development. So two key features enable myogenic PH-LH proteins to promote muscle cell differentiation. So firstly, the basic domain binds to a muscle cell specific enhancer sequence adjacent to genes that are only expressed in muscle cells. And secondly, their activity is regulated by dimerization. So heterodimers may be activating or inhibitory. So in the case of this BHLH protein E, this is the binding of this heterodimer activates this gene, whereas in the case of this this protein ID heterodimer that it's unable to bind to the gene, and so the gene is not activated. Okay, so so this particular heterodimer is inactivating. As this ID protein binds, this BHLH protein can no longer bind, whereas in this case, this one activates it such that it is able to bind and drive expression of that particular gene. So the problem then is that only a process with foresight can visualize a complex end goal and bring together everything that's needed to realize that end goal. And any process with foresight is going to be indistinguishable from intelligent causation. And therefore this, I think, points to an intelligent mind as being the best explanation of these cell lineages culminating in a viable organism. So I will pause there and turn it back over to Dr. Christie.
Thank you, Jonathan, for that wonderful explanation. Next, let me come to another question for you. I would like to ask you about the challenge of metamorphosis. It's a process through which some animals abruptly transform their bodies after birth. This has long inspired misunderstanding and also mysticism. Metamorphosis is a truly bizarre process. Today, as many as we can see, 65% of all animal species on this planet are metamorphosing in sex. Like a butterfly, a fruit fly undergoes 4 stages in its short life cycle, from egg to larva, pupa, followed by remarkable resurrection from inert pupa to a flying adult capable of sexual reproduction. What's interesting is that their life cycle is controlled by a sophisticated interplay among genes, hormones, environmental factors, and checkpoints. For instance, if a fly larva has to reach a critical size to pass from one checkpoint for further development. Example, to survive the pupal stage, when it can't eat, it needs a minimum supply of reserves on hand. Until it reaches that critical stage, development is delayed. That is, till the food supply improves. Once past the checkpoint, though it will enter the pupal stage, reserves or not, it will just end up being a smaller fly. So it's quite amazing. Like us, flies have glands that produce hormones, numerous acceptors, promoters, and receptors that turns the genes on and off. So is it possible to explain this metamorphosis through an unguided process like the evolution? So Jonathan, could you throw some light on that?
Sure thing. Share my screen again here. Okay. So yeah, the challenge of metamorphosis again is a significant challenge to evolution. So there you got 2 animal body plans. You have the caterpillar, and then the caterpillar has to enter at the chrysalis stage, and in the chrysalis, basically its body is decomposed into a type of soup, and then the animal is able to reconstitute itself and produce a new body plan, which looks nothing like a caterpillar. The scale of the transformation is just incredible. I mean, it has to switch from eating plant material to eating nectar, for example. The caterpillar can't reproduce, so only the butterfly has reproductive capability. The changes have to take place in the locomotory system for them. There's such changes have to take place in the antenna and the brain and so forth. There's just so many changes that are needed to transform a caterpillar into a butterfly. And the question is then, how can evolution select for a non-viable intermediate form? Because why would natural selection select for the organism to be basically dead inside this chrysalis without some plan of getting out of that state. And so again, this is going to be a process that requires foresight to realize, to actualize, to bring about. And any process with foresight is going to be indistinguishable from intelligence. So I have a short video animation to show on this, which I think is quite a good summary of the challenge that metamorphosis poses to evolutionary biology.
If you open the work of a lepidopterist, someone who studies butterflies, somewhere in that writing, you're going to find the language of astonishment.
Every one of these 20,000 species have different color patterns, and every one of them has different shaped wings. The diversity is just so magnificent.
As you watch a butterfly, to describe what you're looking at, you can't really put it into words. That's biology, it's also magic.
It's impossible to look at a caterpillar turning into a butterfly and not ask how. Their metamorphosis, their life cycle, how did this happen? This astonishing, remarkable transformation.
In a metamorphic insect, what you've got is two body plans. You have to first form one functional body plan, and then you have to switch gears and you have to take and form a new body plan. I am amazed by development when it goes from egg to caterpillar, because it's such an intricate process. But then you have to enter into the chrysalis stage and you have to get it right again. So it's like the problem squared.
You've got to get rid of or digest the caterpillar tissues. They won't work for the adult. In fact, the cells themselves disappear, but then their components are recycled and are turned into a kind of soup out of which the adult structures will be built. It's very carefully engineered. So you got to know where you're going to end up before you start.
It's like a different organism. Transitions have to take place in the heart. Transitions have to take place in the antennae. Transitions have to take place in the reproductive organs.
You have to recreate adult legs, adult antennae, adult eyes. You have to change the shape of the brain and the connections between the antennae and the eyes. You have to reformat the gut so that it switches from eating plant material to eating nectar. You begin to see the depths of the problem. So for evolution to have created this sort of pathway gradually, it would take a miracle.
When you see certain effects in nature, it's your responsibility as an investigator to find the cause that will explain the effect. If you saw a mechanical device of the sophistication of a butterfly, you would not for a moment hesitate to ascribe that to intelligence, because the butterfly is so much more sophisticated, almost beyond our comprehension, than anything that we make. Planning, foresight, artistry, engineering. When you process all the evidence revealed through metamorphosis, and then you ask yourself, in your own experience, what kind of cause could bring about these results? I think the only reasonable answer is an intelligence that transcends the natural world.
All right, so I will close with that and see if there are any questions on anything we've covered.
So I think, Jonathan, you need to wait for a few more minutes. People, they have some more questions. But my question next is to Prem. So now let me ask two more questions to Prem. We all know that Darwin's theory says that there is a tree of life where you can have one organism diverging into many organisms, and big differences appearing at the top that we can see right now. And his theory postulates that new species are descended from other pre-existing species, and that population of the pre-existing species changed into new species over periods of time. So thus it predicts that there were organisms that existed at transitional stages where one form turned into another. It's possible that evidence of this evolutionary transformations may be found in the fossil record. That's what he wrote in his book, Origin of Species. But he was waiting till the end. That was the one that was so much scary for him. He could never find any transitional forms, or intermediary forms. So now he thought that, okay, over the course of time, when they unearthed a lot of fossils, they might find a lot of transitional forms. But to the horror of many evolutionists, the transitional forms are not as they expected. We will cover that in our next episode. Now, let me come to my questions. As per Darwinian evolution, the whole strata should be filled of transitional forms, but we don't see that. So in 1972, Gould and Eldridge, they proposed the theory of punctuated equilibrium. Under punctuated equilibrium, species usually change little because gradual change is not the normal state of a species. That's what they say. By reducing the numerical size of the transitional population and the number of years for which it exists, punctuated equilibrium greatly limits the number of organisms bearing transitional characteristics. So since many organisms are not fossilized, this increases the likelihood that transitional forms would not be fossilized. So Gould and Eldridge claim that it is predicted by population genetics too. But what are the implications of punctuated equilibrium? Normally, every scientific theory should be testable because they are testable when they make predictions which scientists can in principle observe. Is punctuated equilibrium a scientific theory or not? Would you throw some light on that, Prem?
Yes. So just to go over the idea of punctuated equilibrium again, really what this is, the theory that's being set forth, that was set forth by Eldridge and Gould, is an attempt to explain the lack of transitional forms in the fossil record, which is something that Darwin himself noted in the 10th chapter of Origin of Species, which he referred to as the imperfection of the geological record, which is his own admission that he's not able to find the amount and variety of transitional forms that would be predicted by his theory. And so here we see Eldridge and Gould essentially saying that the fossil record does not seem to show evidence of the kind of gradualism that Darwin expects from his theory, according to which we ought to be able to see numerous transitional forms. Instead, what we see is whenever a species appears in the fossil record, it remains unchanged for most of its time of appearance, and then suddenly appears to undergo rapid change. When we say sudden, we mean in geological time, it's sudden. So in other words, they are acknowledging that the fossil record shows species going through what we call stasis, which means that there is really no change happening for most of the time of its appearance in the fossil record, and then suddenly within a few generations, there is a lot of structural change, morphological change, and so essentially it's saying that evolution doesn't happen gradually; it happens in jumps. So one thing to remember is that one of the critiques of Darwinian the theory of evolution is this notion of the tree of life. So if you look at a biology textbook, you're going to see some kind of a tree structure that and each point of branching, you'll see some type of organism that's posited there. In some cases, these organisms are actually found in the fossil record. In other cases, the organism is posited even though there is no evidence of such an organism, but it's necessary to make the tree look like a tree. And so what we actually find is that there's less and less evidence for there being a tree of life. So the only place where you find a tree of life is not the fossil record, but really within evolutionary biology textbooks. So what we see is the sudden appearance, which we've already covered in the Cambrian explosion. And then Eldridge and Gould, they are also saying that when you examine the fossil record, new species appear rather quickly, and then they stay unchanged for long periods of time. And like anything else that has to do with geological time, of course, punctuated equilibrium is not testable because it requires observation over such large periods of time, which are basically not conducive to human beings. It requires the ability to, if you were to look for it, you'll have to sort of observe the fossil record over millions of years. Jonathan, I don't know if there's something you would like to add on that subject.
No, that was a good summary. One thing I would perhaps add is that though there are sometimes some really neat transitional series, like for instance, I talked last time about the whale series going from land-dwelling terrestrial mammal to a fully aquatic whale. And if you look at the fossil record, that is usually cited as a flagship example of a transition that occurs in the fossil record. The problem, though, is that the time available, the time that's afforded by the fossil record is too short to be reasonably accounted for by the sort of standard textbook account of little changes acted on by natural selection. And so even though I think that I think the whale series is a good example of a transitional series in the fossil record, the time frame that's allowed is far too short for it to be reasonably explained by naturalistic evolutionary processes, if that makes sense.
Yeah, okay, thank you, Prem and Jonathan. The final last question, and then we can go over to the participants for their questions. I would like to ask this question to Prem. Every field of science has unanswered questions. And also we have gaps in our understanding. Scientists typically view these as open research questions. Others sometimes argue that if science cannot explain how something happened, then God must be the explanation. Such arguments are called the God of the gaps arguments. Earlier, people thought lightning is something from God, and so they could never explain the phenomena of lightning. So they thought God is there behind it. So such arguments are called God of the gaps argument. But now this sort of arguments are wrong, and because science is developing every day. And so the gap gets filled and that God gets expelled. That God disappears, I would like to say. So now look at this one. Evolutionists always tell about the creationists that they are using this God of the gaps fallacy. But when I look at the punctuated equilibrium and many other theories they have brought in, I feel that they are committing the same fallacy. So Prem, what would you respond to this question?
Yes, I think that it's a very good question. So let's go over this idea of God of the gaps, because it really is a fallacy. It's not helpful at all to posit divine activity every time there's a gap in our understanding of some material process. And in this subject sort of bridges what happens within the debate on evolution and origins, but also the subject is pertinent to how we understand God and how we understand God's action and God being a cause of things that we see in the world. So in that sense, it becomes relevant for understanding the Christian view of God. So it's a theological, it is of relevance to theology as well as to the current discussion, which really has more to do with origins. So God of the gaps is sort of an attempt to try to find a role for God in a time when it seems that whatever we thought would be attributable only to divine activity is now being slowly explained by natural processes. So example would be, as Dr. Christie mentioned, this idea of lightning. So because we didn't understand how lightning is produced, it seems reasonable to just posit that God must be directly responsible for lightning. And then as our understanding of physics improves, we find that there's perfectly natural explanations for why it is that lightning appears in the sky. Once we understand electromagnetism and how electricity flows, atomic structure, electrons, and so on, we realize that those who said God was responsible for the lightning look rather silly or outright stupid. So we don't want to commit God of the gaps fallacy. So a couple of things here. One is, how does a person who is a person of faith retain a place for God in the scheme of explaining the material world? So here we have to differentiate between immediate causation and ultimate causation. So if we understand that God is the ultimate cause, that is to say, God is the one who causes everything in an ultimate sense by being the creator of all that there is, specifically the laws of nature and the fact that organisms have a certain design, they work a certain way. And therefore, the phenomena that we see, whether it's in physics, whether it has to do with lightning, or whether it has to do with the working of any given animal, that these are all moving according to some initial design and some fixed set of laws. And that God remains in the picture as the author. Coming from a very theistic perspective, God would be the author of the laws of nature and the initial design. So there's no fear that somehow God will become expelled from the picture of the world just because we are able to explain things that we were not able to explain before. And I think the last time we met, we had this discussion about looking at the calculator. So for example, if you look at the calculator, you can ask the question, why is it that when you punch in two numbers and try to add them, you get the correct answer in the calculator? And you could resort to explaining how the calculator works and talk about how you've got wires and you've got LED output and you've got the electrons moving through the wire and so on and so forth. And this serves as some sort of mechanistic explanation for why the calculator works. And that's one kind of explanation, and it would be correct. However, if I were to ask you, why is it that the output is always mathematically correct? What is it that accounts not just for there being some output, but for the fact that the output is always accurate? Then we have to speak about not just the mechanism, but the fact that the calculator has been programmed or designed, and therefore it would require something other than just material. It requires some sort of intelligence to produce the kind of programming that would make the calculator to be accurate. So there's a different explanation, and that's where the positing an intelligent cause comes in. So I think that instead of trying to find a role for God by inserting God as a term in the explanation every time there's a gap in our understanding, it's better to conceive of God as the creator or designer of the laws of nature and the initial design that's inherent in nature, whether that's living things or non-living things, so that God does not lack a role at all. He's definitely the creator, also the sustainer of material physical reality. But what about evolutionists? If you look at the theory of evolution, both with Darwin and today, there is this sort of optimism that the theory of evolution will cover all the gaps in our understanding. And so the problem here is more the presupposition that naturalism is going to be sufficient. In other words, just only using natural causes without using any kind of intelligence as a term in the explanation, that we will be able to explain all phenomena that we see. And so this is clearly not possible. So, but what's happening is that in the narrative, in the evolutionary story or narrative, we are being told to trust in nature as being the final, being sort of a sufficient resource. So an example would be positing transitional forms when we can't really see any evidence for transitional forms, or the tree of life, where we created this branching structure, whereas the fossil record doesn't seem to show a common point of origin from which multiple body plans come out. Instead, what it shows is the sudden appearance of multiple body plans in the Cambrian explosion. So there's a fair bit of storytelling going on to make us believe that even though there is no evidence, we should trust that naturalistic processes are sufficient. So wherever there's a gap, we just posit nature and naturalistic forces, in this case, randomness, really, random mutations, to fulfill the role that God would fulfill in the God of the gaps argument. So whenever you don't understand something, religious people have been accused of saying, well, God did it. So that's an illicit use of giving a role to God whenever we have a gap in our understanding. And so my point is that within evolutionary theory, we're doing the same thing. But instead of saying God did it, we say, well, nature must somehow have done it, because we don't want to allow an intelligent cause, or whether that's God or some other unspecified intelligent cause. So it's more of a resistance, philosophical resistance to the idea that naturalism is not enough. So it's important to understand that in the evolutionary narrative, this fallacy is being committed. So it's not just religious people can fall into that wrong way of thinking, but even those who would espouse evolutionary theory to be able to explain all of life end up having to do that. So we could call that nature of the gaps instead of calling it God of the gaps, which is what religious people are accused of doing. The same thing is happening in the evolutionary narrative, and in a sense, nature is substituting for God, if you will, to explain things we cannot explain.
So thank you, Prem, and thank you, Jonathan. So from this discussion, we could easily infer there is an intelligent design behind everything. And understanding that everything came through an intelligent design is the right approach, is the rational approach. When you think that everything came by chance or an accident, there should be proof behind it. So with this, let us finish this discussion, and let me open up for questions from the participants. You can ask any questions.
I dropped a message to a question to Jonathan, but in brief, the question was about microevolution and how even within a species, like I think you took up Danio zebrafish, and how there's ontogenic changes that are being observed, which is a bit peculiar considering that some of these are like expressed at an younger age, but then they disappear at an older age, like how we saw in the case of metamorphosis in a caterpillar to butterfly. So I was just wondering, microevolution, and maybe that's a whole another topic, but how would that also explain an intelligent being or God?
Sure. So you said you worked on the ontogenic changes observed in the zebrafish, Dino Aereo. What do you mean by ontogenic changes? Do you simply mean information beyond DNA, or what are you referring to there?
These are physical, like the high tail breed frequency. This is just an example, like how in a single species you could observe those changes of how a high tail beat frequency, about like close to 100 hertz when it was a larvae, but then it kind of decreases as it grows older. And that protein, the kind of isoforms that exist also disappear in a way, like they don't, they evolve or they disappear. That is something that I wasn't able to conclude. So I was just using that as an example to talk about, just because you were on the slide that was talking about, I think, changes in different species of early acting developmental mutations. That was when I posed that question.
Sure. So I'm not familiar with high tail beat frequency reduction, so I need to do some reading on that before commenting on what exactly that involves. Yeah, so I'm not sufficiently familiar with that to comment on it.
Okay, so there is another question. What is the scope and extent of error in the methods of dating? See, there are a lot of type of dating. Molecular clock assumes that is always it assumes the tree of life is true, and then it proceeds. And similarly, there are radiometric dating and many other things. I think we would cover it in the next episode or the next to next episode. We didn't cover it this time because of the lack of time. So we would be covering fossils and the dating approaches, everything in another episode. If anyone would like to respond, you can.
In terms of molecular clock dating, which I think you might be referring to, there's a lot of variation in the rate of the molecular clock, so across the tree of life. So I don't think that the molecular clock dating methodology is necessarily the most reliable. So yeah, that's kind of what I'd say on that.
But I think it assumes that the tree of life is true, right?
Correct. Mm-hmm. He says also carbon dating. Yeah, I tend to think carbon dating is reasonably accurate. Obviously, carbon dating is and you can't use carbon dating to date things more than about 60 or so thousand years ago, because of the short half-life of carbon-14. But I think generally speaking, carbon-14 dating is a reasonably accurate approach to dating.
So any other questions?
Like one question, it's a more of a layperson question. How do we know that the evolution has stopped, or like some kind of other evolution maybe going on? It's
Can you tell your name and ask the question?
Yeah. My name is John. I've been listening to Jonathan's presentation. So the question is how do we know that there doesn't exist some other kind of evolution, which may be taking place right now, or what does it? What stops it for not participating in that evolutionary timeline?
I'm sorry, I'm not quite sure what you're saying. You said, what do you mean by other forms of evolution?
Like, why not new organisms now coming? New organism, new cellular organism, new structures coming, which are totally new. So what prevents them?
Well, some of the things that I talked about previously, I think that so changes in Hox genes, for instance, like the antennapedia mutant or the ultrapythorax mutant, for instance, result in body structures arising where they wouldn't normally be present, such as the four-winged fruit fly, or the fruit fly with legs growing out where the antenna should be, and so forth. These are not the anatomical structures, because if you're going to build new anatomical structures, you need fundamentally new information content. And simply modifying Hox genes to affect the expression of genes doesn't generate new genes coding for new structures and that kind of thing.
Yeah, this is Prem here. So Jonathan, perhaps I could just jump in and maybe just redirect a little bit. So I think what you said regarding the inadequacy of mutations in the Hox gene to produce new body plans, would you agree with saying that really even the existing body plans don't seem to be a product of gradual change at the genetic level? So it's
Correct.
not okay. So it's not as if evolution in that sense of new body plans has been taking place. For the question then to be, it has taken place, but why is it not taking place any longer? What you're really saying is that existing body plans could not have emerged by that sort of gradual process.
Exactly. Yeah.
Okay. Thanks.
Yeah, and in fact, Hox genes are only expressed after the body plan. The anterior, posterior, and dorsal ventral axes have already been well established. So mutating Hox genes is not going to be able to generate new body plans because they aren't responsible for body plans to begin with. You're going to have even earlier acting developmental mutations in order for you to get a new body plan. But the problem there is that these are the least tolerant to evolutionary perturbation.
So can we finish it with a prayer? Prem, can you finish it with a prayer?
Sure. Our Father and our God, we thank you for the opportunity, Lord, to look upon your creation and to see the glory that is reflected. Lord, your glory, your wisdom, your power, your intelligence. Lord, we are reminded of John chapter 1, in which it is written, in the beginning was the Word, and all things were made by him, and without him was not made anything that was made. Lord, we thank you that the information has only you as its source in an ultimate sense. We pray for all those, Lord, attending. We ask, oh God, that this material will be useful, that you will show us how to use it and apply it in making a case, a credible case for why there is a creator, Lord, and to show that there is nothing within the physical and biological sciences that would contradict the idea of an intelligent designer. Lord, we thank you for Dr. Christie's efforts. We thank you for Jonathan's efforts, Lord, and for all those behind the scenes who help events like this to materialize. We ask, Lord, your blessing, and we thank you in Jesus' name. Amen.
Amen. Amen. Thank you. Thank you. Thank you, Prem. Thank you, Jonathan. Thank you, dear friends, for joining. So we'll come with the third episode soon. Thank you.
Thank you.
Thank you. Good night.
Thank you.